Bismillahirrahmanirrahim. This is the seventh video on this series of molecular bases of cancer. In this video, the evasion of the cell death will be discussed. These are the references for this video. Cells contain mutations that make it resistant to apoptosis, and these cells evade apoptosis. Apoptosis is a mechanism of suicidal cell death or programmed cell death, in which the cell is broken up into small components, and these components are then efficiently taken up by the macrophages without any activation of the inflammation. There are two pathways for the activation of this apoptosis. One is the extrinsic pathway and another is intrinsic pathway. The extrinsic pathway or death receptor mechanism is triggered by death receptor of the tumor necrosis factor receptor family, such as FAS and their ligands, while the intrinsic mechanism, that is also termed as mitochondrial mechanism, is initiated by various stresses such as absence of growth factor and DNA damage. This intrinsic mechanism is the primary judge of life and death in the cancer cells. There are many stresses that lead to the activation of intrinsic pathway or a mitochondrial mechanism. These are DNA damage, metabolic disturbances, hypoxia, and increased amount of misfolded protein. These stresses are increased manifold when tumor is treated with chemotherapy or radiation. The two mechanisms by which the tumor cell evade the apoptosis mechanism is: one, they disable key components of the intrinsic pathway, and second is they reset the balance of regulatory factor so as to favor cell survival. Before going into the mechanism of evasion of cell death, we will first see how the intrinsic mechanism works. The intrinsic pathway of the apoptosis, when activated, it will lead to permeabilization of the mitochondrial outer membrane that will release cytochrome C molecules into the cytosol and starts the apoptosis. The integrity of the mitochondrial outer membrane is made intact by balance between pro-apoptotic and anti-apoptotic members of BCL protein family. The pro-apoptotic factors are BAX and BAC that directly make holes in the outer mitochondrial membranes, while the BCL2 and BCL-XL are the members of anti-apoptotic protein family, and they block the activity of BAX and BAC. While there is another set of the protein, that is BH3 only protein. The members are BIM, BAD, BED and PUMA, and they block the activity of BCL2 and BCL-XL, that are the anti-apoptotic factors, and they then work for pro-apoptosis. When the cytochrome C is released into cytosol, it binds with apoptosis activating factor 1 that activates caspase 9, and then the executor cascade of the caspases is activated. There is another inhibitor of apoptosis protein, that is IAP, that blocks the activity of caspase 9, and hence it is another check on the intrinsic pathway. How this cell evades the apoptosis or the programmed cell death? It does so by two mechanisms. One is the loss of p53 direct or indirectly the diminished function of p53 by the amplification of mouse double minute 2 or MDM2. As we know that this p53 is in bound form in this cell, it is bind with the MDM2, that is a component of a proteasomal degradation system, that is actually an E3 ligase, and it takes this p53 to the proteasomal degradation system. So when directly or indirectly it prevents the upregulation of the POMA. So the pro-apoptotic function is lost. So another mechanism is the overexpression of BCL2 family, that are the anti-apoptotic factors. So when these are overexpressed, the cell leads to, or the cell the cell is survived. Two examples of this overexpression are follicular lymphoma and chronic lymphocytic leukemia. In follicular lymphoma, that is a B cell tumor, and there is a translocation between chromosome number 14 and 18, and this translocation fuses the BCL2 gene to the immunoglobulin heavy chain. That will result in overabundance of BCL2 that will protect the transformed lymphocyte from apoptosis. So there are more follicular cells, or there are more BCL2 in the follicular lymphoma cells. The another example is chronic lymphocytic leukemia, in which the BCL2 is upregulated due to loss of specific microRNA that normally restrains BCL2 level. Identification of the mechanism of the cell survival in the tumors led to recognition of the therapeutic targets for the development of drug. One of these is the inhibitors of MDM2, which is helpful in the tumor in which the cell survival is due to amplification of MDM2, that is indirectly blocking the effect of p53. When these MDM2s are inhibited, these are excessive and these are inhibited, so the p53 function restores. Similarly, the overexpression of the BCL2 that gave the cell immortality characteristic, it will be blocked by inhibitors of BCL2 family. This is the summary of the events we just discussed. When there is DNA damage due to reactive oxygen species, radiation or chemical, or there is deficiency of growth factor and survival signal, or ER stress or unfolded protein response, that is going to activate the pro-apoptotic factors, and these are BAC and BACs. That will lead to permeabilization of the mitochondrial membrane, outer mitochondrial membrane, and cytochrome is released into the cytosol, and it binds with the apoptosis activating factor 1 that is responsible for activation of caspase 9, and cascade of the intrinsic pathway of apoptosis starts. The caspase 9 is also blocked by inactivator of apoptosis, while the anti-apoptotic factors are BCL2, BCL and MCL. When there is defective response of the p53 either due to mutation of the p53 or due to overexpression of mouse double minute 2 or MDM2, it will lead to survival of the cell. Similarly, the increased expression of pro-survival factor or anti-apoptotic factors such as BCL2, BCL, XL and MCL1 will lead to the survival of the cell.